right, so my name is Rachel. I work as an aquarist at Sea Aquaria, and today we're going to learn about bivalve mollusks and ocean acidification. Just a heads up, in this program today, I'm going to ask some questions, so I want you guys to answer in the chat, so just so we can have a little interaction. So just keep that in mind if you're in full screen. So just make sure you exit the full screen so you can access the chat bar.
Okay, good afternoon, everybody. Welcome to the
There you go. Oh, perfect. Thank you. Yeah, so just make sure if you're in full screen mode that you can access the chat when we have those little activities today. Also, another note that there is a lag of about 7 seconds between me and you, so I may not answer questions right away. Don't worry, I'm not ignoring you. We just got to let the video feed catch up. Just also a reminder that during Science Odyssey, we have a little giveaway going on right now. So to enter, you have to complete one activity from each program that we're doing this week. Take a picture of your activity and then share it on Instagram. So for today's program, you can either do an oyster anatomy puzzle, or you can do the shell shock experiment. So just make sure you tag Sea Aquaria Ocean Education using the hashtag Sea Aquaria Learning. Or you can send us an email of all your activities to education@seaaquaria.org. So you can find all that information on our website, that's seaaquaria.org, along with activity packs for this week's programming. All right, so to start, I'm going to introduce you to 5 different bivalves that we have today. I have them right with me in the basins on my desk. Let's see. So before we introduce any bivalves, we got to figure out what is a bivalve. So bivalves are 2 animals with 2 hard shells. So I have an empty mussel shell here to demonstrate. So bivalves have 2 hard shells made of calcium carbonate, and they are attached by a hinge at the end. So they open up like this. Can anybody guess what kind of bivalves I'm going to show today? Put your guesses down in the chat. So the reason why bivalves have 2 hard shells is to protect their soft inner bodies from predators. And to keep these 2 shells closed, they have a strong abductor muscle inside. So this muscle attaches to both sides of the shells and it keeps it shut. So to keep the shell shut, they flex their muscles really strong and it keeps them safe from the outside world, which is pretty cool. But if they want to open up to feed or for any other reason, they relax their muscle and it opens up. That's pretty cool. So imagine you want to close the door on your house, and each time you wanted to do that, you had to flex your muscle really, really hard the whole time to keep your door shut. And then when you wanted to open it, you can relax. So this is kind of what a mussel or any other bivalve would do to keep their front door shut. So it's a lot of work. So that kind of means the abductor muscle is very powerful. It's so powerful, in fact, that if you look at shells on the beach, if you look on the inside, I don't know if you can see it on camera here, but on the inside, if you run your finger along the inside of a shell, you can kind of feel 2 little divots. And the little divots in there is from the abductor muscle, which is pretty cool. So that's how strong the abductor muscle is. It even has an imprint long after the muscle or the bivalve is gone. Pretty cool, right? All right. So one of the reasons why bivalves might open up their shells is to feed. So bivalves are what we call filter feeders, meaning they take in seawater to take in food to eat. So I'm just going to share a little diagram with you guys. Let's see, get my screen sharing here. So this is a bivalve of some sort. It looks like a clam. So what happens is that seawater comes in through one of these siphons, goes into the bivalve, and it goes into the gills on the inside. So once the seawater gets to the gills, the bivalve picks out bits of food, which is called plankton. Different plankton are microscopic small organisms that float in the water column, and at the gills of the bivalve, they pick out all the little plankton, and that gets transported off to the stomach so they can digest it. What also happens at the gills is that oxygen is picked up so the bivalve can breathe. So once the bivalve is finished with the seawater in its gills, it shoots it back out through another portion of the siphon. So it has two. So one is called the in-current siphon, where it takes in seawater, gets processed in the gills, and then it exits out the ex-current siphon. All right. Do that. Let me stop sharing with you guys. Oh, that works. Perfect. Okay. Good. Oh, did I? There we go. There it is. Sorry about that, guys. First live stream, still kind of working out the bugs. Okay. So that's how a bivalve feeds, and that's one of the reasons it would relax that abductor muscle and open up its two shells to breathe. So because bivalves are filter feeders, they're often found in a lot of estuaries, which is really good because they take in all that brackish kind of sedimenty water, and it filters out all those little bits of sediment to make our waters very clear and clean. So that's why bivalves are really important in estuaries and in our oceans in general, so they can keep the seawater clean. It's very cool. So another reason why a lot of bivalves have, or all bivalves, I should say, have two calcium carbonate hard shells is that they're sedentary, which means they stay in one spot most of their lives, which means they need that hard shell to stay protected, because they can't just get up and walk away. If there's a predator, they can't run away from predators. So they need that hard shell to protect themselves as a defensive mechanism. And also because of sedentary lifestyle, it makes them very vulnerable to ocean acidification, which we're going to talk about a little bit later today. So first, let's move on to our bivalves I have with me today. So now we know what a bivalve is. We've learned about their abductor muscle. We've learned about their two calcium carbonate shells. So now let's meet the specimens I have with me today. So I'm just going to move the camera down to the basins here. Just bear with me. Okay. All right. All right, so here are all the bivalves I have with me today. Pretty exciting. Okay, so let's start off with our first bivalve. I'm going to show you guys some muscles. So the muscles are these dark ones here. I'll kind of lift up one to show you. You've probably seen a lot of these guys if you've gone down to the beach at low tide. So they're this is a blue Pacific mussel. They're quite dark in color. They can get quite big. As you can see, the shell of the mussel I was using earlier is huge. So they can get quite big. This one's pretty big as well. I'd say it's about a medium-sized mussel. So because the mussels live in the intertidal zone, which means they get exposed to the air twice a day, they can be out in the air for quite a long time. How do you think these animals are adapted to surviving out of the water? If you guys want to pop down and answer in the chat and take some guesses, you can do that as well. But yeah, so these guys have to be well adapted for being out of the water twice a day. So to do that, they have their shell to keep all the moisture in, because their inner body's got to stay moist so they can stay alive. Right, okay, so this is the Pacific mussel, and as you can see, it has some tentacles that stay attached to a lot of rocks so they can stay in place. Often they attach to rocks that are in a lot of currents and lots of waves. So they gotta stay attached to the rocks so they don't get washed away. It's pretty important. And they also like to cluster together like this. All the little tentacles. Maybe you guys can see it there. See right in there, those are all little attachments they have to stay attached to the rock. Very cool. There's a little smaller one there too. So we also have two types of oysters here today. So we have Pacific oysters and Olympia oysters. So I'll show you guys both now. Let's see. So we have this big guy. This is a Pacific oyster. And then this guy right here is an Olympia oyster. So you can see there's quite a big size difference between the two. Can anybody guess down in the chat below which of these oysters is the invasive species? Just take a guess. Is it the big Pacific oyster or is it the small Olympia oyster? Put your guesses down in the chat. I'll give you guys a minute. So we're just guessing right now, which oyster do you think is the invasive species? Is it the small Olympia oyster or the big Pacific oyster? Discuss it with your classmates. Give it a little thought and give it a guess down in the chat. Okay, so we have division 2-3 guesses that it's the small oyster. Interesting choice. Okay. Okay, Yuko saying the big Pacific oyster. All right. Those are all really good guesses, guys. So the invasive species is actually the big Pacific oyster. Yeah, so the Pacific oyster was actually brought over from Japan during the gold rush era. So gold was discovered on Vancouver Island. Everybody came rushing over to try to get some gold, but in the process, the Pacific oyster came over with them. So they have taken over because they're so big and they take up a lot of space, which kind of competes out the small Olympia oyster. And these two guys like to live in estuaries where there's a lot of sediment. So the Pacific oyster has a much easier time being out of the sediment, so they don't get buried. Whereas the Olympia oyster is so small, it gets buried very easily. And because the Olympia oyster is further endangered, when all these people came over to pan for gold, they would also eat the tiny Olympia oyster as a snack. So they loved them so much, they ate them all up, and now there's not a lot of them left. So here at World Fisheries Trust, we have a big research project trying to get a healthy population of Olympia oysters back in Victoria waters. So that's pretty cool. I love these guys. These are one of my favorite bivalves. I think they're so gorgeous. Maybe I can get a little closer for you guys so you can kind of see all the colors it has. If the camera would focus. It kind of has like some greens, some browns, even a little bit of blue on the side there. Very pretty. And you can see Olympia oysters like to settle onto even more oysters. So all the shells underneath this guy are oysters that have passed on and they've left their shells behind, but it means it's a new home for living ones. So that's pretty cool. So speaking of Olympia oysters, we're going to take a closer look at what is inside an Olympia oyster with this video. So I'm going to share the screen with you guys real quick. Just bear with me. Share the screen here. All right, perfect. So I'm just going to play this video for you guys. That's going to show the body plan of an Olympia oyster. Oh. Oh, I don't have sound here. Just 1 second. We'll mute that. I think that's gonna work. You guys hear that okay? Hmm. I'm not so sure what's going on.
So we're just experiencing a little bit of a technical difficulty, everyone. Our sound doesn't seem to be working. So give us just one moment. Thanks for your patience. Our technology doesn't seem to want to work with us today. So instead of playing the sound with the video, I'm going to get Rachel. Could you just narrate over the video? I know you had sound with this video, but unfortunately, it doesn't seem to want to cooperate. So I think the next best thing is to narrate over the video as it plays.
Okay, yeah, sorry about that, guys. We're just gonna play this one more time, and I'm going to explain what is going on here. For sure. There we go. Okay, so to start off, for Olympia oyster, it has hard outer shells made of calcium carbonate, and the shells can be many different colors. They can be brown, green, even blue. They're all mottled, and also a lot of algae likes to grow on the inside, so that makes them very colorful. Next part, we have the mantle. So this separates the hard outer body to the soft inner body. And it's usually white or soft pink, and it's ringed with dark tentacles on the outside. So the oyster can sense what's out there. So they're like little sensory tentacles. Next, we have the gills. So they're usually a dark gray in color and U-shaped. And as we learned earlier, they filter out food and what is not food, and they also use the gills to breathe. Next, we have the palps. This is where food gets sorted out once again. So it separates out any sediment or dirt that may have came in with the water. And it's for further processing over to the stomach, which we're going to see next. So this is the intestinal loop. It has a mouth at the beginning, a stomach in the middle, and at the end it has the anus. So all the food is processed here and further digested. Next, we have the digestive gland. This is another organ that digests food even further. And these are the intestinal loop and the digestive gland are both transparent. So they turn the color of the food that the oyster is eating. So for a lot of plankton species, that's green, yellow, or even red. Next, we have the abductor muscle. We have learned about this already. It's a strong muscle that keeps the two shells together. And very last, we have the gonad. So for Olympia oysters, they switch between sexes each spawning season, so they can both produce eggs and sperm. And the color of the gonad varies on the time of year. So it can range from gray to pink to even orange. The very last tiny organ is the oyster's heart. So it doesn't have any color at all because Olympia oyster's blood is colorless. So you can color this heart however you'd like. All right, thanks for watching it one more time with me, guys. So sorry the sound didn't work. It was unfortunate, but that's okay. We persevered. Excellent. Okay, back to the basin. So those are Olympia oysters. And once again, they're native here. So there are only native oysters here on the coast. So that's pretty cool. I love Olympia oysters. I think they're just gorgeous. So just a reminder, you can find that oyster anatomy activity that I showed in the video there in today's program. So that's all on our website at seaquaria.org. So all the bivalves that we've looked at so far have their hard shells and strong abductor muscle to keep them safe. But there's a couple bivalves that have a couple more strategies that use more than just staying sedentary and in their shells. So the next one we're going to look at are clams, and they have a different method of escaping predators. So we can only find one clam in the lab today, and this is him. He's a little banged up, as you can see, but clams are very cool because they burrow themselves into the sand on beaches, and they have a foot that helps them escape. So when they sense predators, they go deeper into the sand with this muscle called a foot. It would come out. He's not going to show his foot today because I'm handling him. But if he was in the sand, he would have a muscle coming out right here, and that's his foot. And that's what he uses to escape from predators. So clams are excellent by escaping by digging deep down into the sand. And there's a lot of different species of clams in BC. There's around 7 or 8 different species. So these animals are quite plentiful here on the coast. And for many animals, it's a huge food source, including humans. So for many Indigenous people along BC's coast, clams are an important food source. Many cultures ate clams right away or stored them away for later use throughout the season. Clams could be baked, steamed, dried, or even roasted. The clamshells were also important. So not only did they just eat the soft insides, they also used the shells. So they were used as spoons and cups or even jewelry. Clams were so important to indigenous cultures, or they're still important to indigenous cultures today, that they even modified beaches into something called clam gardens. So clam gardens are terraced beaches that are to encourage clam growth. So they would block off portions of the beach. So at low tide, the water would stay behind in these big pools or gardens. So the clams really liked that, and they like to grow in the sand in these areas. So for a lot of indigenous cultures, they would have huge food security and would have endless amounts of clams. So that was ensuring food security for a lot of different indigenous people, which is really cool. Yeah, so those are clams. So all the defensive strategies we've learned about bivalves so far is that they have their shells. They can dig down deep into the sand to escape predators. Now this next bivalve is very cool and has a very cool way to escape from predators. And I may be able to get him to escape from a leather star that I have right beside me here. And that is scallops. So I have a big scallop and then I have a small swimming scallop with me today. So these are scallops. This is a big one right here. And so they can attach themselves to rocks or other, oh, just a little squirt at me. They can attach themselves to rocks or other clams via a small little foot right where their hinges are at the back. Here's the small swimming scallop that I have. Here, I might get close for you guys so you can see. So you can see it's very round. They have a lot of grooves right here. It's very bumpy when I rub my finger along here. And they have a very cool ability to escape predators. So what they do when they sense a predator coming, they suck in water through their mouths. So they open up their shells, they suck in water, and they squirt it out the other side where their hinges meet. And they swim away from their predator, which is pretty cool. They also have a lot of different eye spots along the rim of their mantle when they're open. So maybe I'll get to show you guys. I think this guy's just opening up. Might be able, oh, he's closed. Okay, so when the scallops have their shells open, you can look closely and they have lots of tiny little black dots along them. These are eye spots. So they're not like eyes that you and I have. They only detect shadow. So if a predator comes over and puts a shadow over the scallop, their eye spots sense the shadow, they shut tight, and they can swim away if need be. So it's pretty cool. Like, how many bivalves that you know has 50 to 100 eyes? Very cool. So I'm gonna put this scallop in the tank with this leather star. I'll just adjust the camera here really quick. Oh, here, I'm just gonna move this basin real quick here. Get that out of the way. I can get you guys closer. Yeah, I might be able to show you guys. All right, so in here I put the smaller swimming scallop in there. I'm gonna let them adjust. And on the side here, what we have is a leather star. So leather stars are a big predator of bivalves. They're very fast and very aggressive, and they use their tube feet that they have on the underside to latch onto bivalves. Here, I'll show you guys. This guy might look familiar if you joined us for Monday's live stream. We had a lot of starfish and echinoderms in the office. So you can see they have rows of tube feet. They have 5 rows of tube feet that they use to grip onto bivalves and eat them. So they have a stomach that comes out once they open up the shells of the bivalve, break the abductor muscle, and they eat them. Don't worry, I'm not gonna let him eat the swimming scallop. I'm just gonna let the swimming scallop show its swimming abilities. I see he's opened up. Maybe I can show you guys the eye spots they have. Here, I'll put this down. Here, let's take a look. So you guys can see that's the scallop. He's opened up his shell. Look very closely. See the black dots are his eye spots. So that's why he uses to evade predators and swim away. Very cool. Here, I'll put you guys back up here. And we can see if he's gonna swim away. So I'm gonna put the leather star on top here. There we go. It may take him a minute to realize what's going on. I'll get you guys down close in action. Might take a little while for the sea star to understand what's going on too. Might be able to sense it. There we go, that's a good view. So you can really see this leather star's 2 feet, always moving around, always feeling around for something to eat or something to grab onto. See if I do that. See if we can see any eye spots as well. Oh, we could see the scallops kind of moving here. Oh, okay, here he goes, guys. Oh, he's kind of feeling, oh, there he goes. Oh, and he's done. Good job, swimming scallop. Here, I'll put you guys back. So that's a swimming scallop. That's how they evade their predators. And as you can see, it's very effective. He is out of the reach of the leather star. So I'm just going to separate these guys because we don't actually want this leather star to eat the swimming scallop, and put him back in his basin. But yeah, so those are all the bivalves that I have for you guys today. Very cool. Give those guys a round of applause. Good job. Okay, guys, so I'm going to switch gears and we are going to move on to ocean acidification. So I'm just going to share a PDF with you guys real quick. Share the screen. Okay, so you can also find this PDF in the package on the website as well if you want to take a quick look at it after the program, if you want to refer back to it. So we're going to do our ocean acidification experiment pretty soon, but before we do all of that, we need to know what ocean acidification is. So ocean acidification is when carbon dioxide from cars or any fossil fuels that have been burning is absorbed into the ocean, and it creates a chemical reaction that with the seawater, and as a result, it makes the seawater more acidic. So a solution that is acidic means that it is corrosive. Does anybody know what corrosive is? Does anybody know what the word corrosive means? You can pop any guesses you have down in the chat, but corrosive means that it eats away and destroys most things. So if you're in very acidic water, it's going to eat away at your skin or any shells in the water is going to disintegrate. So it's very destructive, anything that is corrosive. So that's what that corrosive means. So if you're an oyster in the ocean where ocean acidification was happening, it wouldn't be very fun for you, especially for bivalves, because they can't grow their shells properly in the water that is too acidic. So, but how can we tell if water has become acidic? Does it look different? Does it smell different? So this is where the pH scale comes in. So the pH scale is a scale numbered from 0 to 14, and it tells us if a solution is acidic or not. So when something is not acidic, we say that thing is basic. So 0 would be very acidic, and 14 would mean that not acidic at all or basic. So a good example of something that's very acidic is vinegar. I have on the diagram here. So vinegar has a pH of 2. And something that would be very basic would be something like bleach, which has a pH of 12. And something that's neutral or is not too acidic but not too basic is 7. And that would be something like drinking water. So now that we know a little bit more about ocean acidification and the pH scale, let's practice our knowledge a little bit. So I'm going to play a fun game with you guys. So this is where, if you're in full screen mode, you might want to exit out and access the chat bar. And we're going to just practice our knowledge about the pH scale here. So hold up, maybe I'll stop sharing for a quick sec. Get this ready, bear with me. Okay, so you guys ready? I'm just going to share my screen now. So this is the first thing we're going to look at. So this is our first household item that I'm going to put onto the screen. And you guys got to tell me in the chat if you think this item is acidic or basic. So here we go. So first one we have is oranges. Do you guys think oranges is acidic or is it basic? And also bonus points if you guys want to take a stab at what the number on the pH scale would be for oranges. Okay, so we got a class saying it's acidic. Okay, good guess. I'm just gonna wait a couple more seconds just to see if anybody's got some last minute answers they want to put in. Oh, we got Yuko saying acidic. Okay, well, we got some very, very smart people in the chat today because you're right. Oranges are acidic. They're actually very acidic. So if we think back to the pH scale when we were talking about acidic and basic things, oranges are 3 to 4 on the pH scale. So those are pretty low numbers, which means low numbers means something is quite acidic. So that was good, guys. That was the first good round. So let's do this one. Okay, what do you think, guys? Do you think toothpaste is basic or acidic? This one might be a little tricky. It's hard to tell when I'm thinking about when I brush my teeth with toothpaste, do I think if it's acidic or basic? Ooh, okay, we're saying basic. Okay, well, you guys are correct. So toothpaste is basic. It's around an 8 or a 9 on a pH scale. So that's pretty basic. That's pretty above 7. So next one, let's do this guy. So what do we think? Do we think soap is acidic or basic? So this might be a little tricky one because we're not tasting soap. Hopefully not. We're just using it on our hands and stuff. So we got to think about it on our hands. Is it feeling acidic or basic? All right. Okay, we're saying basic again. Yeah, that is correct. So soap is basic. So it sounds like you guys got the hang of it, but I have one last one to go, and this one might be a little tricky. I know this one kind of fooled me when I was first learning about it. So let's see. Guys are pH masters. So what do we think eggs are? Do you think eggs are acidic or basic? All right, any last minute guesses for eggs? Acidic or basic? Okay, Yuko's saying acidic for eggs. Anybody else wants to contribute? Okay, votes are for basic. Okay, so we got two conflicting guesses here, acidic or basic. Okay, grand reveal for eggs. Eggs are acidic. Yes, eggs are acidic. So they're not too acidic. There are 6 on the pH scale. So if you think back to when we were saying 7 was neutral because it's right in the middle of 0 and 14, eggs are just like a little bit acidic. They're a little, just 1 away from 7. Okay, guys, that was great. Thank you for interacting with me and playing that game. That was really good. You guys are masters of the pH scale. Okay, so now that we know all about the pH scale, ocean acidification, bivalves, abductor mussels, I think we're ready to move on to our experiment. So this experiment is called Shell Shocked, and we're going to experiment, or we're going to study the effects of ocean acidification on bivalve shells. So I'm just going to stop this share screen right now so you guys can see the lovely experiment. in front of me. Here, let's see if we can get a better angle for you guys. Might have to move down here. Sorry, just bear with me for 1 second. I'm gonna get all set up for our experiment here. Move these over. That's a little better. You guys can see it. All right. So this experiment is called shell shock. And once again, you can find the whole write-up for this experiment that you guys can do on your own time. So once again, we're going to study the effects of ocean acidification on bivalve shells. So when scientists do an experiment, what they need to follow is this method called the scientific method. So I have a little diagram right here to show you guys. Here, let's see if it's gonna work for you. There we go. So in the scientific method, don't worry, we've done most of these steps already. We come up with a problem or a question that we have. We gather information about that problem and or question, and we study it and make sure like what the problem is about. And then we make a hypothesis about that question and create an experiment to figure out the problem or the question that we have. So we're at this part of the scientific method where we're going to experiment to test our hypothesis. So what you're gonna need for this experiment is 3 different containers, and we're gonna label them with vinegar. We're gonna do half vinegar, half water. And then we have a control in the experiment where it's just gonna be plain tap water. And then you guys are also gonna need several bivalve shells. Doesn't really matter what shells they are. They could be scallops, mussels, clams. You can go down to the beach, collect some shells. Just any shells you'd like. We're gonna need those. We're gonna put them in each of these solutions here. And you're gonna need some plain water. I just have some in a mug right here. This is just plain tap water. And then you guys, we're gonna need an acid. So we got some vinegar, so pure white vinegar. White is fine. And what we're gonna do, we're gonna put shells in each of these containers. So we're in the vinegar container. I think I'm gonna put maybe a scallop shell, maybe a clam shell. Yeah, let's just do 2 for the vinegar. Let's do that. Now, before we put the shells into the container, it's important to take observations throughout the whole experiment. So when we look back on the experiment, we can see what happened as our results come in. We're like, how did that happen? I was like, well, this happened during the experiment, so maybe that explains how we got this result. So just take note of your shells, like what ridges are on them, what color they are, what they feel like, even their weight. Like just, you might have to write it down and see and compare your notes after the experiment. So these ones, the scallops are quite rough, while the clams are quite smooth. And it feels like the clam's heavier than the scallop. So we're gonna put those in there. And you do that for all the shells going into all your containers. I'm gonna put that in. I'm gonna put, I'm not quite sure what this looks like an oyster shell. We're gonna put that in the water. I'm gonna put all those in. So in our vinegar container, as you can guess, we're just gonna put straight vinegar. So I'm just gonna put this in here. You don't need to fill up the container all the way, just enough so you just cover the shells. No need to waste all the vinegar. So we got vinegar in there. So now in the half water, half vinegar, so you guessed we're gonna put half water, half vinegar. So I'm gonna pour just about half in here. There we go. And we're gonna put half water. So I have some water right here. There we go, just enough to cover the shells, perfect. And then as you can guess, in the water container, we're just gonna put straight water. So we're gonna fill this until the shells are covered. Perfect, okay, so we got all our containers going. So another thing to note during the experiment is just take a peek into the containers right when you put the solution in and see how do they all look different from each other. So if we take a look in the vinegar one, you can see there's a lot of bubbles going on, right? And then this one, a lot less bubbles. And then this one, in the plain water one, looks pretty calm. Okay, so interesting. So make sure throughout the experiment you note any changes or differences between all the containers, and leave these overnight for 24 hours. And when you revisit in the morning, see how your shells have changed. Has the vinegar, the plain vinegar one, seem less degraded than the water one, or is it the half vinegar, half water that's going to be the most degraded? Just take note of how the shells are different after 24 hours. Okay, so this is an experiment you guys can do on your own time with your class. It's a lot of fun. You also don't have to use just vinegar. You can do lemon juice, you can do malt vinegar or apple cider vinegar. And you can also experiment with different types of bivalve shells. So it doesn't have to be just scallops or clams, or you can note differences, how the mussel shell degrade better or worse than the scallop shells, just stuff like that. There's a lot of cool things you can do with this experiment. So this experiment is supposed to represent what happens when bivalves are in ocean acidification waters. So maybe the vinegar represents some water that has been through a lot of ocean acidification, or the half vinegar, half water is just some ocean acidification. So this just shows you a real life example of what's going on with bivalves when they're in the ocean and ocean acidification is going on. Right. So that's all I have for you guys. Here, let me just switch this over to see better. There we go. Excellent. So that's it for our program today, guys. Thank you for spending your afternoon with me. I really appreciate it. Thank you for going through the technical difficulties we had today. It's good. I have a little bit of extra time to answer any questions you guys have, or if you want to see any bivalves again real quick. You can drop those in the chat box below. Let me know. Looks like the scallop is slightly open again. There we go. I'll just play a little bit so the video can catch up. All right. Okay, so if that's it for questions, I think I'll let you guys go. So thank you again for watching. I hope you guys learned something today about bivalves and ocean acidification. Make sure you tune in the same time tomorrow For the last day of Science Odyssey, Kayla and I are going to take you on a virtual walk of the Gorge Park here in Victoria, BC. We're going to explore some of the local plant species, and my personal favorites, we're going to look at some local bird species. I love birds, so this is very exciting. Tomorrow's program for me. So come join us tomorrow. Have a lovely afternoon, and we'll see you tomorrow. Bye, guys.